I am Michele Curatolo, University of Washington in Seattle, and I'm happy to share perspective for a better targeted pain treatment. The typical pathway to drug development is to rely primarily on animal studies and then translate the findings to human pain. Unfortunately, only very few medications have survived the process of drug development for use in patients. And we will discuss the reasons for that and potential solutions in the following slide. While a lot is known about molecular mechanisms of pain in animals, much less has been studied in humans, mostly because it is much more difficult to study deeply the mechanisms of pain in humans. Obviously, this limited knowledge has hampered the development of really target-specific therapeutics. As a result, as many of you know, because some of you may be patients, current therapeutics do not really work great, offer at best modest benefits. Typically, molecular targets are discovered first in animal studies. Then, based on those studies, drugs are developed that are then studied in patients with pain. Also, the efficacy of these drugs can be studied in healthy volunteers by inducing pain with different methods, for example, heat, pressure, and other kinds of stimuli. However, these pathways did not yield so far very satisfactory results. Why is that? Well, recent studies have found substantial differences in molecular mechanisms of pain between animals and humans. And also, experimental methods to induce pain in healthy volunteers frequently do not reflect the pain experienced by patients. We now have new ways to study pain in humans. The approach that I will discuss involves first the discovery of mechanisms primarily in humans. Then we can validate the results in animals, for example, by testing medications in animals after making sure that the mechanisms are comparable. Finally, we proceed to studies in patients with pain. So probably this pathway is more likely to lead to effective therapeutics of pain because the discoveries primarily are made in humans. So I'm presenting two studies that we have just started. In this one, we are studying patients with low back pain who undergo spine surgery anyway. We will collect during surgery different tissues, nerves, intervertebral disc, facet joint, muscles, and fascia, and examine molecules that trigger the pain signal using advanced methods called RNA sequencing. We will associate the molecular findings with different pain measures, such as the severity of pain, disability, and other outcomes. Now, because pain affects mood and other psychosocial functions, we will have to account for these factors in our analysis. That's very important. This way, we hope to identify molecular targets of therapeutics that will be more effective than the treatments we currently have. Another study will be on low back pain and fibromyalgia, and we will use so-called metabolomics. Metabolomics is the study of the thousands of metabolites produced in the body and can be measured from blood and urine samples. So we will compare metabolites from patients with metabolites from pain-free subjects. And again, we will associate the findings of those metabolites with different pain measures like I explained for the previous study. Again, we hope to identify pain-relevant molecular pathways that will facilitate the development of better therapeutics. Finally, I would like to stress that pain is not only about molecules. It is a complex, multidimensional experience. It is very unlikely that any medication will be sufficient in most patients. In the context of integrative medicine, it is essential to consider other elements of treatment, such as lifestyle, nutrition, physical activity, and mental health. So that's the end of my presentation, and I would like to acknowledge the collaboration with the University of Texas at Dallas, MD Anderson Cancer Center, and the collaboration with the National Institute of Health and Merck. Thank you for your attention.